Hi, this is Valentina Pacella and I'm happy to present OHBM 2023, the study on the exploration of the fabric of the human mind. Most of what we know about the brain-cognition organization has been derived from fMRI studies. And the reason why we love fMRI is that it links task-based activation to a specific cognitive construct. However, if some functions seems to be segregated in the brain, others seem to rely on common brain structures. Moreover, the recursive validation of experiments and theoretical conceptualization of cognitive construct hinders the exploration of new cognitive functions that are still encountered in the clinical practice. In this study, we want to explore the latent organization of the brain-cognition relationship. And we wanted to quantify the degree of reliability of the cognitive functions studied by fMRI so far. Moreover, we wanted to provide a predictive framework to explore new cognitive functions that still haven't been identified. We aim to do so by leveraging the meta-analytic approach. Meta-analysis allows to summarize the results from individual studies and provides new insights that are not immediately visible. So we use the Neurosynth meta-analytic repository that computes coordinate-based functional meta-analysis maps related to specific cognitive terms, and we selected 506 cognitive terms that are related to 506 meta-analytic maps. We parcel those maps and we reduce the dimensionality of our data via the UMAP algorithm that allows to project and reduce the dimensionality while retaining its core organization. So we obtained this 3D space with the embedded meta-analytic maps. And the maps organize themselves in the space according to their similarities or differences. More specifically, maps that are similar cluster close together, while maps that are different spread far away apart. And in the 2D visualization of the space, as you can see in the right, we can observe that the space has a neuron-shaped architecture. And we were able to identify the branches of the neuron-specific cognitive domain, while the center of the neuron is unspecified. To explore what's going on in the center and to quantify the degree of reliability of the functional meta-analytic maps explored so far, we built the rationality index. The logic behind it is that to predict the morphology of a territory in a specific coordinate, we will leverage on the morphology of the nearby territories. And the resulting prediction will be more similar to the closer territories and more different than the distant territories. The same applies to our space, meaning that if we want to predict the anatomy of the finger movement maps, we will rely on the morphology of the anatomy of the other maps in the space. However, if the information of the morphology of nearby territories is corrupted, this bias will be reflected onto our prediction. And the same applies to our space, meaning that if we are leveraging studies that have no control task or have been poorly theorized, this bias will reflect onto the anatomy prediction of our map. To quantify the relationship between the maps, we computed Euclidean distances between each and the other maps of the space. And we use these Euclidean distances as independent variable in as many models as the number of our maps. Our rationality index will be then the correlation between the original and predicted map. The results are shown in this figure, where we can see maps with low rationality index in red and maps with high rationality index in yellow. More specifically, an example could be the listening map, and you can see visually that we were able to predict the meta-analytic map, while for other maps, such as the consciousness map, the prediction is really poor, meaning that the rationality index is very low. And if we zoom in on the language branch, we can see other examples of maps that have a high rationality index, such as semantics or lexic. And example of maps that have low rationality index are common umbrella terms defined maps, such as executive functions, or more inner states related maps, such as feelings. We explore also the inter-hemispheric differences in terms of rationality index, and we confirm the literature by identifying that the left hemisphere has been more rationally studied than the right. Then we wanted to explore the most rationally brain structures. And to do so, we use the rationality index of all the maps in a regression model. And we obtained highly rational structures, such as superior temporal gyrus, inferior frontal gyrus, pre- and post-central gyri, hippocampus, basal ganglia, and supplementary motor area. We then correlated our map of most rationally explored structure with the maps of the gradient that explains the human cognition in a data-driven way, that have been studied by Margulies and colleagues in 2016. And we saw that our map highly correlates with the gradient 1 and 2, meaning that our structure that have been most rationally studied are actually unimodal brain structures, with some exceptions, such as supplementary motor area or inferior frontal gyrus, that are more higher order cognition related structures. We then wanted to explore whether we are able to predict new anatomy of new cognitive functions in the gaps of the space. So we used new meta-analytic maps that are not included in the brain cognition space, that are derived from NeuroQuery and Neurosynth, and single fMRI results from single fMRI studies derived from Neurovault. We parceled the maps and we projected the maps onto our space. Then we computed Euclidean distances between the new maps and the other maps of the brain cognition space, and to compute the rationality index of the new maps. The results show that maps actually locate themselves in the gaps between the branches, and maps that locate in the gaps between the branches and closer to the branches have high rationality index, while maps that collocate onto the center of the space have actually low rationality index. Finally, we wanted to test whether we are able to predict mental states with our brain cognition space. And to do so, we used the HCP dataset, and we collected more than 3,000 maps related to the movie dataset. We parceled the maps and we projected them onto the brain cognition space. The results show that we are actually able to predict the thoughts and mental states of the participants while observing the movie. And for instance, the figures show that brain activation related to each frame of the movie collocates themselves closer to specific maps of the brain cognition space. To see an extended example of the frames and the brain cognition space maps related to that frame, please check the link in the comments. In conclusion, we were able to unveil the latent organization of the brain-cognition relationship that has a neuron-shaped architecture. And we also quantified the degree of rationality of the thus far explored functions by fMRI. Finally, we provide a predictive framework to explore the anatomy of new cognitive functions and to test the reliability of new fMRI studies. I want to thank my collaborators and thank you for your attention.